In nature, animals compete to survive. The fittest, fastest, strongest or smartest will make it. And for most of human history, we too have struggled to survive. Finding food, fighting disease and dealing with the environment around us have all been great challenges. But humans have overcome many of these. Our population has exploded as we've built cities, farmed land and developed cures for most of the major diseases of the past. Today, humans have the understanding and technology to alter the basic structures of life, the genetic code of any living organism. We could ensure every baby is healthy or make crops grow in the desert. But how is this possible and what precautions do we need to take? In this program, we look at the science behind the benefits of genetic engineering, including genetic manipulation, the basics, genetic manipulation, what, why and how, human uses of genetic manipulation, agricultural uses of genetic manipulation, environmental uses of genetic manipulation, and ethical issues. You probably know genes provide the information for all cells, tissues and organs that make up our bodies. They're our blueprint. We could say genes are the instruction book for all the proteins that make up our structure. Our genes are made up of DNA. That's deoxyribonucleic acid. You might recognise the DNA double helix. It's structured like a ladder. The rungs of the ladder are the 4 nucleotides. They link together like this. Adenine links to thymine, cytosine links to guanine. These base pairs line up in a specific order. It's this order that determines which protein the gene codes for. The process of reading the DNA instructions and building the protein they describe is called protein synthesis. It starts in the nucleus where the DNA is untwisted, then read and copied into a strand of messenger RNA, or mRNA. We call this transcription. It's important to remember that the information in a gene is read in lots of 3 bases. Each triplet of bases is called a codon. Each codon is the information for a specific amino acid. The mRNA moves out into the cell where the codons are translated into their matching amino acid. These are linked together to form a protein. So that's how DNA works as the body's blueprint. So what would happen if we could take out a segment of the DNA and change the function of a gene? We'd be changing a chapter in the instruction book. That's called gene manipulation. Before scientists can change the genetic makeup of an organism, they need to understand which genes carry which information. So let's take a closer look at a gene and see how that's done. A gene is made up of a promoter, a coding section, and then a stop codon. The promoter marks the start of the gene. It's like the on-off switch. And at the end of the gene is the stop codon. This gives the instruction to stop making the protein. Some sections of the DNA are non-coding. We also call them junk DNA. These sections don't carry any instructions for making proteins. Here's a coding section of DNA. It's our gene. This section carries information. If scientists can figure out what a gene codes for, then they have the option of changing that information. Genes can be moved, copied, or even put into other organisms. More about that in a moment. Genes provide the instructions for all the proteins in our bodies and are made up of DNA. Every animal and plant has its own DNA. DNA has coding and non-coding sections. Scientists can change the genetic code of an organism if they know which genes code for which information. So now we have the lowdown on genes, it's time to get to the bottom of genetic manipulation. You've seen some dramatic results of genetic technologies on TV. Dolly the sheep, Gene therapies? Maybe one day a cure for cancer. But how does all this happen? Nature often makes mistakes in copying a gene's DNA sequence. The result of this is a change in the genetic code. We call it a mutation. Some mutations are harmful, but some are helpful. Genetic manipulation allows scientists to decide which genes to change. They can carefully alter the DNA sequence, changing the gene and the protein it codes for. So let's say we have two organisms. We'll call them A and B. And we want to take a gene from A and put it into B. Scientists use a technique called polymerase chain reaction, PCR, to copy and study genes. Let's take the DNA from our organism and copy it. PCR allows scientists to make many copies of the gene so they have enough to work with. The next step is to take this section of DNA and place it into a second organism. We can do this using a vector. Scientists sometimes use viruses as vectors. The viruses aren't infectious and can be packaged up with our extra piece of DNA. The viral vector takes this new genetic material and places it inside our target cell. Or here's another way to do it. The gene can also be incorporated into a little ring of DNA called a plasmid. This DNA ring is taken into the foreign cell and incorporated into the genetic code. Those few steps form the basis of genetic modification. Of course, when you talk about genetic engineering, most people think of cloning. To clone means to copy. We can clone a piece of DNA to put it into another organism. This is called molecular cloning. We could clone a cell to make many copies of it. This is called cellular cloning. But what about making a copy of a whole organism? For this, scientists use a technique known as nuclear cloning, or somatic cell nuclear transfer, SCNT. They take the nucleus of a normal body cell, which contains all its DNA, and put it into an egg cell, which has had its own nucleus removed. So the egg cell has its DNA replaced, and the idea is that the new egg cell will develop into an organism with identical DNA to the original. You've probably heard of Dolly the sheep. She was the first animal to be cloned this way. Of course, this isn't just science fiction. Genetic modification and cloning are being used for all sorts of things right now. We often refer to this as biotechnology. Biotechnology is technology based on biology. Up next, we visit scientists in their labs to see some biotechnology in action. So, genetic modification is based on the idea of copying a gene and placing it inside another organism or cell. By copying genes and attaching them to a vector, new genes can be added to an organism's genetic code. Scientists can also clone genes or organisms. One of the most exciting areas of genetic research applies to the health and well-being of humans. So how can doctors use genetic manipulation? Imagine being able to cure a disease by changing a person's genetic code. Already, scientists can identify the genes that cause some diseases and then work on changing those genes. Heidi Peters is a doctor and scientist at the Royal Children's Hospital in Melbourne. She works with children who have an unusual problem with their livers.
So I work on a condition called methylmalonic aciduria, and that's a disorder of organic acid metabolism. It's inherited, it's rare, and it's a problem with the breakdown of certain, with the breakdown of protein really, and certain amino acids. So you have an enzyme that's missing, and that enzyme causes methylmalonic acid to be built up in the body and causes the patient to become unwell. I look after patients with rare metabolic disorders, and as part of that, I guess, you become frustrated that there's no new novel therapies for these patients, and you have a very limited amount of treatment available. And I guess that's sort of driven me to want to look at development of novel therapies to try and help treat these patients.
So Heidi works with children who have MMA, and she also does research in the lab to try to find a treatment for the disease. The first part of her research involves transplanting healthy liver cells into mice.
So the mice are a model for the condition that I'm interested in, so the methylmalonic aciduria. And these mice are transgenic mice, and they have a phenotype, or they have characteristics that would be similar to a mild form of the MMA condition.
So these mice have had a gene inserted that gives them a mild form of MMA. They're transgenic. Heidi's team transplants healthy cells into the mice. The transplanted cells produce the missing enzyme, so that's one way MMA could be treated. The next step is to genetically change the cells before they're transplanted.
So one way is to take the cells for transplant, treat them with a viral gene therapy vector or a non-viral approach, and so that they overexpress the gene of interest, so in this case the MMA gene, and then to transplant those enhanced cells into the liver so that each cell would be more effective than if you just transplanted non-enhanced cells. The thing that's exciting for gene therapy is that the potential to improve patients' health, really.
Gene therapy is the insertion of a gene into an individual's cells. The idea is that hereditary diseases can be treated by adding a new, healthy gene. Heidi's team are planning to treat healthy liver cells with a gene that will make them produce even more of the missing enzyme. Then, they'll put the cells into the patients. Well, mice at this stage.
So with viral gene therapy, you are putting in, using a virus to deliver a gene of interest that may not be working properly, or you want to overexpress or essentially into a cell or into a body or into an organ, so that it can then start functioning. So that's one form of gene therapy. What I enjoy about the work is that it gives me the opportunity to be able to sort of study something that might be able to be of benefit for treating patients. I think really that's what drives me. The aim would be to improve their life, essentially, to reduce the burden of the disease on that patient and that family. Hopefully give them a better quality of life and a longer life.
So, you can see there are already some exciting medical uses of genetic modification. Gene therapy could also treat diseases like muscular dystrophy or cystic fibrosis, and there's a lot more research going on around the world. There's also xenotransplantation. That's when animals are used to grow human organs. Many people die each year because of a lack of donor organs, but transgenic pigs could provide a good source of human-like organs that wouldn't be rejected by human immune systems. Scientists can also use biotechnology to create proteins in the laboratory for medical use. The insulin diabetics need to survive used to be sourced from pigs. Thanks to biotechnology, insulin is now made in a laboratory by inserting artificial genes into bacteria. The bacteria then produce the vital insulin needed to help keep people with diabetes alive. These proteins are called recombinant proteins. This technology already has a wide range of medical and scientific uses. There are a number of medical uses of genetic manipulation. Gene therapy involves adding a gene to a cell in order to treat an inherited condition. The gene can then produce a protein that was previously missing or faulty in that cell. Other techniques now being researched include creating human proteins using modified bacteria or animals, and xenotransplantation. Of course, genetics doesn't just apply to humans. All animals and plants have DNA. Genetic modification also offers exciting ways to protect animals from disease. Let's take a look inside the CSIRO's Australian Animal Health Labs. Kathleen McLaughlin is a PhD student there.
My research is in developing better ways to protect livestock from viruses which cause disease, and the way that I've been doing this is to use a new technology called RNA interference. The major benefit of RNA interference is that you can specifically silence genes.
Kathleen is working on finding ways to stop disease in farm animals. In her lab, they're studying something called RNA interference. To put it simply, they're using the natural response of cells against viruses to silence genes.
Gene silencing is when you switch off a gene, so you have a gene that's normally active and we call that switched on, and you use RNA interference to silence the gene or switch it off. So if you think of it like a dimmer style light switch, when the gene's normally active, the light is at its brightest. When you use RNA interference to silence the gene, you decrease protein production, and it's like turning down the dimmer light switch. And then if you completely silence the gene and there's no protein production, it's like the light is switched off and the gene's silenced.
So by turning off or silencing a gene within the virus, Kathleen's team is working to prevent the virus from infecting animal cells. They're trying to block the conversion of RNA to protein for a particular gene that the virus needs to infect cells. Right now they're working on protecting chickens, but who knows what kind of vaccines this research might lead to. Agricultural science is a really important area of research. It's all about making farm animals and crops stronger and healthier to create food for more people. It's now possible to select desirable characteristics for a crop. So maybe you need a plant that grows in salty soil or very cold weather. Scientists can identify and transfer genes that make a plant resistant to certain conditions into another crop. These genes are transferred to vectors that can transfer into the plant's cells. We call these transgenic plants. Transgenic organisms are those that have a gene transferred from another species. In nature, genes are sometimes passed between species, but this happens over a long, long time. In the laboratory, it's a much speedier process. Transgenic crops can be engineered to be pest resistant. It's also possible to create transgenic animals by transferring a gene from one species to another. Transgenic cows can have a gene inserted to make them produce more milk or milk with less cholesterol. There are transgenic sheep that produce more wool and cattle that produce better meat. Scientists are using these technologies to figure out ways to increase farmers' yields and profits. The hope is that they'll be able to feed more people using less land, less water and fewer chemicals. Genetic manipulation can allow scientists to grow stronger, bigger and healthier crops and animals. Scientists can transfer genes from one species to another. This produces transgenic crops and animals. They can select genes that make crops grow in difficult conditions. They can also use genetic manipulation to protect crops and animals from disease or pests. It's not just people in industry that benefit from genetic manipulation. Genetic technologies can also play a big role in keeping our environment safe and clean. We've already seen how transgenic crops can help farmers grow more food in harsh conditions, but this technology can also be used to conserve sensitive environments. Dr. John Patterson has good reason to be interested in helping farmers and the environment.
So I grew up in the country on a farm in East Gippsland and had always wanted to get into agricultural research.
Today, John is working to create crops that can survive in areas that are dry and salty.
The main goals of our research are to develop crops with improved tolerances to different environmental stresses, so things like drought and salinity, which are major issues for Australian farmers. The work that I do focuses on looking at the proteins and how the proteins in different plants change in response to different stresses. If we can identify proteins that allow a plant to survive in a dry or a saline environment, we can use that information to develop improved varieties that might have expressed that protein or that gene, and use that to improve the tolerance of other plants.
John identifies plants that are tolerant to dry or salty conditions. He compares the genome of these to plants that can't survive in the same dry, salty conditions. By doing this, John can figure out which genes might be the ones that make a plant tolerant to drought or salinity.
The next step, once we identify these genes, is to put them into intolerant varieties and see if they have any effect on improving the drought tolerance of those plants. So the way we do that is through a process of transgenesis, where we actually insert the genes that we think are interesting into these intolerant varieties and look at their effects. So these types of approaches are looking very promising, and we are in the process of developing some crops with some important agricultural traits that will have benefits for Australian farmers. The research that I've talked about today does have a number of positive impacts on the environment as well as the economic benefits. By creating varieties of crops that have improved drought tolerance or improved salinity tolerance, we can grow more crop in less area of land, so we can use less land for agricultural productivity, and also we use less water to grow the same amount of crops. I really hope that we can identify some genes and then use this information to develop some varieties with really improved water use efficiency and drought tolerance, and get that research out into the field so that farmers can actually use this information.
John's work has economic, social and environmental impacts, and he's not alone. Scientists around the world are working on similar genetic technologies. Many industries produce pollution that finds its way into our waterways, our air and our soil. Biotechnology is finding ways to clean up contaminated areas. One involves phytoremediation. Phytoremediation is a way of cleaning up pollution using plants that are able to break down polluting chemicals. Biotechnology allows scientists to find genes from these species and put them into another species better suited to the cleanup site. So cleaning up oil spills in the ocean, for example, is becoming much faster and more effective. This technology, called bioremediation, uses genetically modified bacteria to clean up toxic spills or turn waste into less harmful chemicals. Another big problem in natural environments is invasive species. Genetic technologies are already being tested on preventing the spread of some pests. Daughterless carp and toads have been developed in Australian laboratories. By genetically modifying the pest species so they can't reproduce, their environmental impact can be dramatically reduced. Genetic manipulation can play a role in conserving our environment. Work on drought and salinity tolerant crops can help the agriculture industry and the environment. For many people in the community, genetic manipulation offers new and exciting technology, medical miracles, or better environmental protection. But new science and technology always comes with fears. Is it really okay to change the genetic code? Who should decide what the limits to genetic engineering are? It can be a very emotional topic. Like all new science, researchers work hard to test these technologies in the laboratory before they are used out in the real world. Many people have concerns about the risks of changing the genes of crops. They're concerned about long-term health risks and also about effects on biodiversity. One major concern is cross-pollination. Since the pollen of some plants can be spread so widely by wind, insects or birds, it's difficult to control how plants interbreed. If the pollen of a genetically modified plant is spread to normal plants, the new characteristic will appear in other plants too. This isn't always a problem, but it is important to consider what the long-term effects might be. For example, what if we make some crops resistant to herbicide and this trait spreads to nearby weeds?
Some people think that the risks of genetic manipulation are too great. Others say it's unethical and that humans shouldn't play God. Ethics and safety are an important consideration in biotechnology.
With genetic manipulation, it's important to consider all the outcomes and to test the technology well before it's applied.
But problems arise when people disagree on the ethics of a new technology. For example, we all know it's unfair for athletes to cheat, and that's why competitions like the Olympics have rules against using drugs to enhance performance. But consider gene doping. That's when genes or genetic material are used to improve a person's athletic performance.
At the moment, it's impossible to test for gene doping. It doesn't involve using drugs, but is it ethical?
Other questions about the ethics of genetic manipulation focus on the potential of parents to select characteristics of their baby. If this were possible, would some traits disappear? And is it ethical? If gene therapy and other treatments become widely available, will they be provided to everyone? Or will people with more money be the only ones who can extend their good health and their lives?
Genetic manipulation has great potential to improve our lives for the better, but there are definitely some big questions to be asked along the way.
It's important to consider the ethics and all possible outcomes of using new technologies and treatments. Scientists work to test the impacts of any genetic modification before it's used outside the lab. One concern is that humans shouldn't be playing God by changing their genome, or that this technology will not be available equally to everyone.
Genetic manipulation is a very recent science, and we're still in the early stages of figuring out how it can be used.
In this program, we've seen how manipulating the genetic code can have applications in human, agricultural, and environmental health and science. This technology also has economic and social impacts, so the possibilities of genetic manipulation are extremely far-reaching.
By understanding the way genetic manipulation works, as well as its potential, we can all play an informed role in making decisions about the future of science.